How's it going? It's March 22nd. It's a little after 10 o'clock at night. This has become the project of all projects. I coined it today on a podcast. I called it the viral defense database. What I hope this Microsoft Excel file and program and algorithm, genetic, molecular, biomedical, biomolecular genetic Excel application is going to supposedly do here when it's done is identify the composition of, in this case, viruses, because we're working on COVID-19 right now, identifying how they're constructed, and then try to find a molecular defense against it or a molecular attack mechanism, something to pry apart or destroy or bond to whatever this virus has on it. So I've been studying up on my molecular biology and my chemistry, and these are areas that I was probably pretty bad at in school. So I'm paying for it now. Got to learn all this stuff. And I'm definitely learning how to code this in Microsoft Excel and build a database of everything. So let's talk about what we've learned over the last couple days. Been working on genes and been working on amino acids. Amino acids are fun. There's 20 of them. They are 3-character chains of RNA, and they have names, these amino acids, and they have a state. And that's what I learned about the last few days, which is they can be either relatively acidic and have a low pH. They can be relatively basic and have a higher pH than the pH of 7 in the body. They can be hydrophobic, meaning they fear water or I guess resist water, or they can be polar with no charge. And then there's these ones that are at the end or beginning of chains that don't have any information on them yet. Point is I was able to figure out what these states are and learn how an acidic amino acid or a basic amino acid relates to what's going on in your cells. One is kind of shedding an electron or shedding an element, and the other one is kind of bonding to or plus sign or going after an element. They're amine groups and carboxyl groups. Fun stuff that it takes a scientist to learn this. But what I'm going to show you is how I've incorporated this into the table of the virus. So I've got the virus and I've got the genes that are in the virus in order. There's like 29,000 base pairs or nucleotides or whatever you want to call these things, molecule bases inside the RNA of the virus. Now, what's cool about this now is that I have this state that they're in is you can say, well, the structure of this entire thing, what kind of states are the different areas of the genome in, or even what kind of states is the whole thing in? And the answer is, well, as I was saying, there's hydrophobic, basic, polar, acidic, and nothing on the end, which I got to identify. So, well, basically what this is saying is that almost half of this thing is made up of hydrophobic amino acids. That's probably what gives it surface, right? Because if it's trying to avoid the water and circulate through your system, it probably has to be kind of like waterproof on the outside. So half of it is hydrophobic. And these are the hydrophobic amino acids that are comprising this. Um, you can see that it's, well, I don't want to say predominantly leucine, but a lot of it's leucine. And then there's valine, and there's isoleucine, and phenylalanine, and alanine, and glycine. These are the hydrophobic elements and the percentage of this whole thing they are. You can also do this by genes. The one gene that I want to go after, because it's the biggest gene, is this ORF1AB gene. This ORF1AB gene seems to be like probably a lot of the structure of the virus, I would imagine. And it's got this, 78% of it is either hydrophobic or polar materials. Like what's the breakdown of the actual amino acids that are in there? It's still predominantly leucine or whatever. Um, and beginning, I don't know exactly what all this means. And also like the order matters too. So when you want to grab the order of everything and see how it's copied, you can see that the different states in order might kind of look like this. You can see that it's very hydrophobic and polar at the beginning. Then there's a little bit of basic, then a lot of hydrophobic, a lot of hydrophobic, basic, polar. This is probably what creates a structure, and there are going to be three-dimensional structures that this thing is eventually going to mimic and generate so that we can understand what this order means in terms of natural structure. And it's just making me think that there's going to be a way in the future here to understand when you have a lot of a certain amino acid in a virus. In this case, it is leucine and valine. I mean, that stuff's going to exist in other parts of your cells all over the place. Um, there's going to be a way to find different chemicals that are going to either bond or react or destroy the chains that are created, the polypeptides that are created by these combinations. So this is quite interesting, learning about all these different states that these amino acids are in and how they structure together and burn things or combine to form the bonds when they move water out, their shape of them. There's a math to this that has already been discovered, I'm sure, by molecular biologists and doctors and everything who've already looked into this. They probably haven't looked at it this way in Microsoft Excel just because they probably haven't. They've used other different software applications which would enable you to browse through genomes in a way that you would want to, but not optimal. And I feel like I can colorize this and try to figure out what the orders of these really mean in terms of the protein shape so that instead of seeing this as just the name popping down, the name and the state, that we're really going to be able to have a whole nother different column called like, horseshoe protein that locks in, like hydrophobic horseshoe. And when you've got hydrophobic horseshoe, you either want to pierce it right down the middle with something that is not water-based because this thing's hydrophobic, or you're going to want to latch onto both ends and pull the horseshoe with you. Like there are going to be different ways that these physical structures can be mitigated as we fight this virus with everything we can. Because every time I look at the numbers, I just see that it is go time, go time, go time. Why don't we have some type of breathable treatment so that we can bond to these viruses and particles as they are floating around in our lungs? We've got to do something about it. We're 7.5 minutes in. We understand that when you look at, I was messing around with this, looking at this in so many different ways, trying to get some information from this. Let's go back up to the order of the, or this is the order of the genome, the genes in order. Just try to look at, what's the distribution. The colors are going to mean, like, just look at like hydrophobic stuff. Your hydrophobic stuff is not on the ends and not on the beginnings. It's like more basic on the beginnings. Polarity. Just to see the change in the colors and the change in the weights in each one of the different genes. This is all going to mean stuff about shapes. I just don't know what it is yet, but it's definitely going to get there. So uh, feeling really good about the direction this is going. And tomorrow's homework is going to be combining, like trying to look at that each one of the amino acids and amino acid chains and finding some qualities that are related to them. Because right now, when you look at your amino acid chains, you, you don't necessarily, kind of when you look at all this in regular order, have this all broken down in codon order. This is going to give you what the gene is and its state. Oh, that's going to blow up. So, like, you got this start of this isoleucine and then lysine and then glycine, leucine, tyrosine, leucine. There's all this leucine up here. Well, is there any way to disrupt this leucine, which seems to be bonding things together? But this is just the 5' end at the top. Well, what about the ORF1AB? How does that start? That starts with an initiator, which is methionine. It's hydrophobic. Then we got an acid. Then we got a polar. Then we got a whole bunch of hydrophobics in a row. Um, but they're different amino acids, and you can click on these and see what their 3-character designations are, or the triplet would actually open up if we just open this up over to the right. you'll start to see kind of combinations of which triplets always appear. Like one of the base pairs will always be in the middle for like a power triplet. Let's see if this works. I'm also going to be able to compare different genomes of the viruses that we find all over the world to see if they're mutating and where they are mutating. It's really easy to compare this through the table. So these hydrophobics, right? These hydrophobics, they like to have a thiamine or a uracil if it's mRNA in the middle or to the right. Or middle or to the right if you've got a T that likes those hydrophobics here in the middle of orb 1. Then you've got more adenines in the polar acid, basic, hydrophobic. It's just weird how you get a group of a bunch of hydrophobics and then you get a group of stuff that is not hydrophobic. So this stuff is combined together to form a string that's trying to get away from the water. So maybe it's going to loop over on itself. This stuff, not as much, or the acid's going to shoot away from something and the basic is going to want to bond to something that it can find free floating. So it's going to move in a certain direction. More hydrophobic chunks. There's more acid in this part. It's going to come out to be like a graph and a color of all these different things. I haven't got there yet, but I just see it developing into something. So I like the direction we're going, but it's clearly complicated as hell. And we'll just keep working on it. So if you're interested in helping me with this, or you know something that I don't about how to get more information that's relevant here, please let me know because we're on the clock, right? Economy doesn't start until we get a treatment cure for this. So that's what we should all be doing right now. All right, good luck. See you all tomorrow.